I think most people are familiar with them. And just in case you didn't get the notice, I am a professor from Ohio State University. So when you see all these names, I'm associated with Steve's lab and the great people in the lab, but I also use Ohio State as my email. Uh, let's see. Okay. Yeah, so here we go. The other islands that you might not be as familiar with are Chappaquiddick, which everyone knows the name, but it's right here. It's actually part of Martha's Vineyard, and Tuckernuck Island. So I'll be featuring the talk. And a little background about why I became interested in ticks. My background is mostly academic background is in academic areas of plant ecology and engineering. I have you guys down on the screen. But in 2014, I was in a workshop sponsored by NSF at MIT, and that was when Kevin Esfelt and George Church talked about gene drives. It's the first time I had heard about them. And shortly after that, they published a paper in 2014 in the eLife about how gene drives might be a new way to look at genetic engineering wild species for applications in human health, environment, and agriculture. And specifically, there's interest in doing this with mice. So here's a real overview of gene drives in case you're not familiar with them. There's kinds of gene drives. One can be for population replacement, like changing a host for a Lyme pathogen into one that cannot sustain that pathogen, just changing that throughout the population. There's another type of suppression, which would, for example, over here on the right, instead of just having all the mice get the new trait, you could have all of them be male, and then you would be suppressing a species that you didn't want. So these kinds of applications got a lot of attention in around right after, and this on the left, this report, Gene Drives on the Horizon by the National Academies, to a lot of really good background about the science and the and what applications are likely to move forward and when. So one of these that you might be familiar with is Isaac Tick's project, which Sam Telford is involved in, and most people on this call would know Sam Telford and Kevin Esfelt at MIT and Joanna Buckthorn. So they described what they thought would be a good application of gene. They started giving talks on Martha's Vineyard. This bottom left slide is from a talk to the Board of Selectmen in Nantucket back in 2018. So I got very interested in this, and a little bit more background about what they're proposing. And this is from their paper in 2019, is to start genetically engineering mice on uninhabited islands, and introduce them onto Nantucket and Martha's Vineyard, and then eventually onto with a gene drive. So starting without a gene drive and adding that later if people wanted to see it. This was some more information that they decided would be phase one, to engineer the mice and prepare by getting baseline data, working on releasing large numbers of mice on these other islands without the gene drive. So they get talks on this. I got very interested in it, and I published a paper in Bioscience just talking about an ecological perspective on their plan. So I'm not going to talk about that, but that's why I got interested. Meanwhile, their research has kind of gone a lot more slowly than they should. So this is a paper in bioRxiv right now that just came out in 2023. So basically, they're delayed, harder to engineer the mice than they anticipated, and the first transgenic mouse was reported in this 2020 goal. And some of their work is also in patent applications. So it's been delayed. So instead of being like a 5 to 10 year project, it's more like we don't know how long this might take. but they have succeeded in genetically engineering white-footed mice in the lab. While I think a baseline data is going to be very helpful, not just for that project, because that might go on, might be very delayed, but for all the reasons we know, the public health reasons. What I'd like to talk about more today is the goal of these tick surveys on islands has been to characterize current conditions and information to be used in any further monitoring. And I'm going to briefly review the work that we've already done on Nantucket and Tuckernuck in 2020 through 2022. And that is summarized in this paper that Steve sent out that's available, published in 2023. And then I'll talk about the work on Chappaquiddick, which we call Chappie for short. And that's work that I started the field portion of it last summer and going to continue again this summer. And there's a progress report available on my Ohio State website summarizing that work. So some additional questions, though, doing these surveys. One of the big questions I have is, are black-legged nymphs still common at wooded sites where lone stars are abundant? And what tends to happen, I think, is when lone stars move into an area, they're so abundant that people have the idea that the black-legged nymphs might be not as abundant. But we don't have a lot of good data on that. And if anybody here knows more about published examples, I'd love to hear about it. In searching around, I found this paper from 2019, Sanchez Vicente working on Long Island, where they just speculated basically that if the expansion of Amblyomma americana continues, it could cause the displacement of black-legged ticks, Axodes scapularis, but they say through competitive interactions that are not understood. And everybody I've talked to can't really think of a way that that might happen, but I think it's a question that we need to ask, both because maybe there are changes in relative abundance or just the perception that people have that, oh, lone stars are so common on Long Island or wherever they're moving in, that maybe the black-legged ticks aren't as common. And then a second question that I'll show you some data on is the relative abundance in the same small area on Chappaquiddick of lone stars versus in different habitats, so open, edge, and wooded. So those are some initial questions. So these surveys that I'm going to tell you about involve all of these species of ticks, and you're all familiar with which ones are common in early summer. And then we're starting to find the larvae of the lone star ticks fairly common too, even though their peak is later in the summer. The chart on the left is from Tom Mather's group from the tick encounter data, and it's a really helpful chart showing peak abundances there of the different ticks. And so if you look at the month of June, it pretty well captures the peak, except for the dog ticks start earlier. And that reflects what I've seen, these general patterns. Okay, so let's see. And as most people here know, the peak of occurrence of Lyme disease and babesiosis and anaplasmosis, it coincides with the black-legged nymphs being out. These are data from Nantucket showing that these diseases are most commonly picked up in late June and July. Further reason to focus on the black-legged nymphs. So in the surveys, I use the same field methods all the time, and I did a lot of reading about different ways to quantify density of nymphs and density of infected nymphs, and basically came up with this plan to work well for just a field crew of one person who has a whole month to spend doing this. So I basically spent the whole month of June and late May as well doing these surveys. So first, select the sites, find trails that have either low vegetation or leaf litter, and if they're in the woods, if they have leaf litter for dragging. I would drag along at least a half a kilometer, so oftentimes finding trails that were longer than that I could use, and stopping every 12 meters to check for ticks. checking each site 4 to 5 days with several days in between in May and June. And on the right, you can see my little field tools that I use. So I like to use this silicone putty when I only have a few nymphs. I take them off the cloth and put them onto duct tape. And the duct tape is in a frisbee. And they generally don't crawl off. And I keep track of them and count them and take them back to my little field station. So I count the number of ticks per kilometer of trail and save the Ixodes ticks for possible pathogen testing later. These on the bottom are some maps of some of my trails. So at the UMass field station, I only had about half a kilometer. And this is a site where Sam Telford has done a lot of work. So everything that I found at that site should be relevant to his publications. Stump Pond was a longer trail. These are all on Nantucket. And South Pasture, that's a pretty long one too, you can see. So the study sites that I chose on Nantucket and Tuckernuck are shown here. The green dots are the ones that were sampled all 3 years for tick densities. The red ovals show where the most black-legged nymphs were found over on this part of Nantucket and also on Tuckernuck Island. And oops, that wasn't supposed to. And then these Lone Star symbols, there's a lot of Lone Stars on Tuckernuck, as I'll be describing. They're starting to appear on the western part of Nantucket. So I added these two sites, which I won't really be talking about today, but they're very common now in these areas of Nantucket. So to quickly summarize 3 years of data, this is the density of black-legged nymphs on Nantucket, averaging from 4 sample days per site. And I've arranged them from the ones with the most black-legged nymphs from Stump Pond, UMass Field Station, Norwood Farm, Jewel Pond. These have a lot. This is nymphs per kilometer of trail. Down to the least, where Linda Loring has the least. And that's the most open. And the habitat differences among these sites are really what you would expect to find, where pine woods have fewer, and these nice shady sites with a lot of leaf litter and often have wetlands nearby had the most. And then you can see a peak in 2021. There were a lot more ticks at several of these sites than in the other 2 years. And then over here on the far right is Tuckernuck Island, which has a lot of Lone Stars. And it showed a huge increase in 2021. It seemed to be different. And Tuckernuck's smaller. It has only 4 mammal species, one of them being the white-footed mouse. So anyways, Tuckernuck has a lot of nymphs as well, and similar to some of these high-density sites for Nantucket. And it's a similar habitat to those sites as well. So those are the density data. I'm going to briefly show you some of the infection data from Steve's whole lab group. And we have data from 2020 and 2021 for quite a lot of nymphs, between 300 and 458 nymphs per site. And the reason for collecting so many was to get these 95% confidence intervals so that we could tell the difference between like 14% and 24%. The confidence intervals don't overlap. So we learned that oftentimes there's not a lot of difference among sites, but occasionally there is. So you can see that for Borrelia and Babesia, the infection levels tended to be around 15 to 25%, depending on the year. And there's more variation between the years than there is within the years across these sites. And they kind of show a similar pattern. And there were more than a random number of co-infections between these two pathogens, as many other people have shown. The other ones that we looked at were Anaplasma and Borrelia miyamotoi. And the Anaplasma was interesting. It didn't show the difference between years. We did not distinguish between the human infective variant versus the other one that is found, that is thought to be hosted by deer, and the human infective one by mice. We didn't distinguish between those. So I want to compare those now real quick to Tuckernuck. So what you can see here is the mean for Nantucket on the left, the mean of the 4 sites, versus Tuckernuck, which showed an even bigger response in the infection levels between the 2 years, and really high for anaplasma, which I thought was strange and might indicate that there's more mice involved in that high level of anaplasma, but it's not something that we looked into further. But these just give you a ballpark of infection levels in the ticks, and they generally agree with other data that Sam Telford has collected on both Nantucket and Martha's Vineyard. Maybe at this point, does anybody have a question? Let me see if I can look at you better. I have plenty to talk about, but if anyone wants to ask a question real quick, you're welcome to. Okay, I'm going to shrink you down. Nolan, maybe if you see anybody raising a hand or asking a question that I don't see, let me know. I don't know if you would see that, but okay. So I want to talk about Lone Stars now and their abundance on Tuckernuck and Nantucket before we move to the vineyard. So here are the data on Tuckernuck. And on Tuckernuck Island, very small island, only a few summer homes. It's about a half a kilometer away from, or a kilometer, I would say, from Nantucket. I sampled along a lot of trail, 1.65 kilometers of trail, but only one area because it's so small. I basically was sampling right in the center of the island right there. And here you can see that the Lone Stars showed the same increase in 2021 that the black-legged nymphs did. And they're very, very abundant. They're more abundant than the black-legged nymphs, but black-legged nymphs are still there. I don't know if this is a trend here where 2022 that I didn't find as many in the blue there of the black-legged nymphs. But it gives you an idea of how incredibly abundant the Lone Stars are. It makes it hard to work on Tuckernuck. They're very aggressive and hungry there. And here on the right, you can see on a lint roller, there are a lot of larvae as well. So all the life stages were present during the month of June on Tuckernuck. So it's kind of weird that Tuckernuck is right next to Nantucket, but Nantucket didn't have very many when I first started. So here at Linda Loring, which is on the western end of Nantucket, this is the trail that was sampled. And these are the raw data from each year of sampling. And then in 2023, Sarah Boyce, who works for the Linda Loring, she's an ecologist there, got data. She's going to continue to monitor this. So they went from being a site with no black-legged nymphs whatsoever to having more and more Lone Star nymphs and adults. And so overall, here's Linda Loring right up here. These are the other sites. It seems really clear now that Lone Stars are starting to invade Nantucket, and they've been on Tuckernuck for over 10 years, and now they're finally starting to spread. And other ecologists on the island are going to be sampling in the same way that I've done, so we can track that change. People on Nantucket are starting to get alpha-gal, and they're starting to notice them more and more. So now I'd like to switch over to Chappaquiddick and tell you why I decided that would be another good spot to study, to do these surveys. And Chappaquiddick has a lot of Lone Stars, and I'll show you some really great data that Patrick Roden Reynolds and Dick Johnson have collected from yard surveys on the vineyard. But here's where it's located. It is connected to the vineyard sometimes, so all the mammals that are on the vineyard can get over onto Chappaquiddick. There's a gap right here where you have to take the ferry. So it's a manageable area to study, and it's got Lone Stars and black-legged nymphs and dog ticks on it. So people on Chappaquiddick actually have approached Steve and other people multiple times saying, is there anything we can do about our ticks? You know, the people who live there in the summer, they're really concerned about all the ticks that they have. So this is a map that that Dick Johnson and Patrick now works in Dick's place. Dick's retired. But they've been studying ticks in yard surveys since 2011. And this is just summarizing their early data. And this is a map of the deer ticks, so the black-legged ticks. And there's a lot on Chappaquiddick, but also really high densities in these areas that have more of the mesic mixed hardwood forest, and as well as oaks along here. So that's what the deer ticks look like. Now, there's several good maps of the invasion of the Lone Stars. And the Lone Stars on the vineyard, they're first noticed in around 1985. There are some articles in the Vineyard Gazette about people noticing them. On Chappaquiddick, they became really noticeable around 2015. And also on this area called Aquina, which used to be called Gay Head, has a lot of Lone Stars. So in all these green dots in the center are yard surveys where no Lone Stars were found. And these are yard surveys. We could spend a whole hour talking about them, but people are invited to request a yard survey. And Dick and then Patrick would go out to do the survey and talk with the people. And they always would do dragging around the perimeter of the property to get these kinds of data. So in 2019, moving forward, you can see there's a lot more concentration of Lone Stars in the two areas, Chappaquiddick and Aquina. And a few more starting to show up inside and more into the interior and other parts of Martha's Vineyard. This is the state forest in the middle, so we're never going to get data on that because that's not part of the yard survey area. That's 2019. And let's see, here's 2023. And this is Lone Stars, many more places showing up, many more yard surveys, and sometimes at very high densities. And the scale on the top right here has changed. It used to be more than 25, and now it's 51 to 100. So it appears that the Lone Stars are going to spread from these peripheral areas into many other parts of the vineyard. And they're also on Cape Cod. So the surveys that I did last summer, I want to summarize now and show you how these were done. The first thing was to look at where are the public properties where there's trails that you could go back to year after year. And Chappaquiddick has a lot of them. And all of these agencies were very interested in having us work on their property and get data that they could use with their management goals. So I ended up choosing 11 study sites last summer, and there are 5 in open habitats, which are shown in yellow, 1 edge habitat right up here in orange, which I'll show you a picture in a second, and then 5 in the woods. And I did this because I knew that the Lone Stars were going to be in a lot of different habitats, and I wanted to figure out a way to quantify that. And just to look at this map a little bit more, these are mostly oak forests where it's browner, and these have higher amounts of pitch pine where it's a dark green. Down here at Wasque, this is a big burned area that is managed by fire. It has no ticks whatsoever. And there's a breach in the barrier island right here that was open last summer. So very strong currents moving across, but now it's closed up again. So Chappie's a small place. My trails were a little bit smaller there, but I found it a very great place to do this kind of research, and the people there were extremely helpful and welcoming to have us working there. So these are what the habitats look like, the open areas where I would drag, like overlapping the taller vegetation as I went through the area. Here's the edge, the single edge site, which is just perfect for deer walking up and down. And so there's a lot of open grass over to the right on this one edge site. And the woods were perfect tick habitat with a lot of leaf litter and a low understory of ericaceous shrubs, and usually different species of oak and sassafras in the woods. So I'm going to show you several slides now where you can see the different sites along the bottom, and they're in order from the open sites the edge one in the middle, and then the wood sites. And this is ticks per half a kilometer. A lot of my trails were only like half a kilometer on Chappaquiddick. And it's the average of 5 dates for each site. So as you would expect, the black-legged nymphs were concentrated in the woods and were not as common in the other areas. And the Lone Star ticks were in a much broader range of sites. And so here you can see the adults are in gray and the nymphs are in orange. Again, this is the number per half kilometer, averaged over 5 different dates. And one of the wood sites did not have as many ticks, and I don't know why, if there were fewer deer at that site or there was maybe a little less leaf litter. What stood out to me is this edge site is really high in Lone Star ticks, and that's very similar to the yard surveys that Patrick and Dick have been doing. So it's just another way of looking at how common Lone Stars can be outside the woods. There weren't very many black-legged nymphs here, but a lot of Lone Stars, and they can also be pretty common in other open areas. And then at these different sites, I found larvae of Lone Stars at 2 of them. Okay, and then the dog ticks are only in the open areas, and sometimes very, very high densities that you need. I always needed a lint roller to sample the dog ticks. There were so many of them. It's all of the adults, and especially at Cove Meadow, there were huge numbers of them. And they were really rare in the woods, even though I know they're associated with skunks, and I saw skunks in the woods, but I saw a lot of skunks over in the open areas too. And I kind of think that might be one of the reasons why they're so common. So if you put them all together, all the sites and the different dog ticks, Lone Star nymphs, Lone Star adults, and black-legged nymphs, you can see the densities can be quite high, especially in the woods. And more than half of the ticks that I sampled in the woods were black-legged nymphs, and these are the Lone Stars here. So they're persisting quite well. I don't know if they were ever more common than this, but that's where they are right now. So that's one way of looking at the question of how well do they persist after Lone Stars move in. So another way of looking at that question is I looked at the average density of black-legged nymphs just in the wooded sites that were on these different islands. So on Chappaquiddick, the average across those sites that I showed you, the 5 sites, was 64 per half a kilometer this year. And this isn't a perfect comparison because these are different years, but it's not that different from Nantucket, which has no Lone Stars. Or just has them on the western end, not in any of the woods. So the wooded sites on Nantucket, this is the densities that I was measuring of the Lone Stars. Tuckernuck bounced around a lot more. These are black-legged nymphs. And Tuckernuck, I don't really trust these numbers because they're so variable from year to year. So the point being that the black-legged nymphs on Chappaquiddick had similar densities to Nantucket in previous years. And then looking at Lone Stars on Chappaquiddick for the nymphs and the adults, these densities were lower than what I had recorded on Tuckernuck in previous years. Just as a point of reference, not to make a lot out of that, but you can't, if you're on Chappaquiddick, you don't tell people, oh, your Lone Stars aren't that bad because they don't want to hear that. They are pretty bad, but they're just not as high as what I was seeing on Tuckernuck. So let's see. So just to kind of summarize the big picture, the habitats, it was very clear that on Chappaquiddick, the black-legged nymphs were mostly in the woods, a little bit, tiny bit at the edge. The Lone Stars were common in edges and also more open areas, both the nymphs and adults. I didn't see any differences in their patterns, and the dog ticks were in the open areas. Okay, and if you look at the different islands, you can see that with Chappie on the right here, it's just a way to look at what's present and how common it is. So dog ticks were only on Chappaquiddick and the rest of Martha's Vineyard, which Patrick and Dick have the data on that. And an interesting difference among these islands is that shown up at the top here. So the number of terrestrial mammals and the area of the islands, and here this large area of kilometers squared is for the whole of Martha's Vineyard. There's only 4 mammals on Tuckernuck. So there's meadow voles and white-footed mice, cottontail rabbits, and deer. So that supports these species, but there are skunks and raccoons on the vineyard, and I think that's the main reason why we don't see dog ticks on either Nantucket or Tuckernuck. Dog ticks used to be on Nantucket a long time ago, and they've become more and more rare, and I didn't find a single one on my trails, but I know they're not quite extinct there. But the lone stars on Nantucket are definitely going to be increasing in frequency just because they're new arrivals there. So that's what I want to cover. I want to save some time for questions. I want to mention acknowledging the different sources of funding that we had, especially Steve's lab, but also other sources, all the access to trails, all the people who helped with this work. It's been really interesting work. And then just to tell you about coming up in 2024, I'm planning to repeat the same field work on Chappaquiddick as before. And also we're working on a manuscript to summarize these data in the journal Insects. We're working with Patrick on combining the yard data, which are really amazing data, over a long period of time with the intensive surveys that we've been doing. So I think I might stop sharing my screen here and see if we have any comments. And I can go back to things if we want to, but does anybody have any comments or questions? Oh, yeah, Andrew.
Hey, Allison, incredible amount of work. You must have spent weeks outside.
It's fun. I like it.
Yeah. In terms of the competition between the black-legged ticks and the lone stars, do you think it's just a limited number of blood meal sources, or there's actually like ecological?
I don't even know if they're competing. That was someone else who made that little statement. And it's hard to imagine that on, deer, I don't know, like the ears of the deer, where would there be any kind of limitation? So it could be competition. It could just be they're not changing. I think what it looks like is that the black-legged nymphs have their own ups and downs, and you just add lone stars to that. And I don't think they're really interacting, but I only have these islands to go on, so I don't really know. I don't think.
Do you have any thoughts about why 2021 was such an outlier in terms of the densities?
Well, yeah, we had higher densities of both species, and it was on Tuckernuck and Nantucket. So I think it was probably some kind of weather patterns that affected, everything upstream of that. Probably the mouse populations. And I'm hoping that now this work started during COVID, which was really bad. My first field season was 2020. So a lot of people aren't publishing much data from 2020, but I'm hoping by putting this out, people will look at those longer patterns and they'll be able to use these data in some big analysis that's more regional. But I think I heard anecdotally that all over New England, 2021 was a big year for black-legged ticks.
Is there any routine rodent sampling on the islands?
Not too much. Sam Telford does some, but I wouldn't say it's very quantitative or useful for looking at these kinds of patterns. He has his other reasons for doing it. So it would be really nice if we had the rodent sampling to go with it.
Yeah, I mean, if it was in synchrony, then it would help at least explain.
But I know. And I really wanted to know more, but I just thought, well, we'll just put it out there.
A whole nother level of logistics.
Exactly.
Yeah. All right, great.
Thank you. Nita, you're muted.
Thank you. Thank you for that presentation. I had a thought about 2021 and I just wanted to bring it up because in 2021 here, it was a decent year. It wasn't like banner year, and I think in Connecticut, especially in this side of Connecticut, in the western part, we haven't been really mirroring what's happening in more northern and eastern New England. In the last like decade, it's kind of split. We're more like New York and New Jersey, but in 2021, we actually saw that our like peak in the black-legged tick nymphs was much later. It was because we sample weekly, and we found that it was actually like the second week of July. And so I was just wondering if you sample like when you sample, you said like 4 times. So I was just wondering, like, do you sample into July, and maybe
That's a
just
really
you might have
important.
just missed it. That's all I was thinking.
Yeah, yeah. No, so for practical reasons and the cost of an Airbnb and things like that, which you don't want to know, I only am there for like 5 weeks. And so I'm hoping to catch the peak. I look at trend. I haven't seen any trends across those 5 weeks of the nymph. There's enough other variation that you don't see that, oh yeah, they're going up more now or whatever. I don't think I could detect it. Um, Patrick Roden Reynolds has done season long sampling on the vineyard, and we're looking at his data too. So there is a chance, and we said this in our paper, that maybe the window of time that we're looking at is not catching the exact peak each year. So, yeah, but thank you for making those points. Yeah, Lars.
Yeah, Allison, that was a lot of interesting stuff. I just wanted to ask you a question about the transgenic mice. Seeing that the mice are refractory to Lyme disease spirochetes only, do you think there would be a change in people's willingness to accept the transgenic mice when we now have a reservoir targeted vaccine for mice on the market, and there's looks like there's a human Lyme disease vaccine coming? Is that going to be a major issue?
I would think
Or
that would probably decrease people's excitedness about having genetically engineered mice added to their environment, but I mean, it's hard to say because I really feel like, from a logical standpoint, you need every tool in the toolbox, and we always want to try different, everything, because no one technique is going to solve the problem. But I think you're right that the acceptability would probably be affected by if the vaccine becomes widely available soon. Is that what you're getting at? Is people might not be as excited to have these mice?
yeah, I mean, yeah, I was thinking so since if the mice were refractory to all the Ixodoscapularis borne pathogens, it would make
That
it
would be a
more
lot.
acceptable. But I mean, I think people would be concerned about just if you release these genetically engineered mice that are refractory to Borrelia burgdorferi, how does that impact all other pathogens they're exposed to? So, and if you put the gene drive in, you can't pull them back. So,
Exactly.
you know,
That's what I was
there's
writing about in that
yeah,
bioscience paper. And I mean, there are new kinds of gene drives that are sort of self-destructing, or they have different names for them, but they're called daisy drives, and they're
right.
not supposed to last forever. These are all being tested a lot in mosquitoes and in lab environments. And so I think for this application, it's going to be 10 or 20 years out, and it's too early to say what people might want before there's anything. I don't know. But I mean, they're looking at, my understanding is they have a lot more funding from NIH now, and it's a long-term project. And this is like a more of an academic project than an applied project right now, just to sort of see, is this even doable? And if it is, what other species and what other diseases might be used in this context? But I can't really speak for them. I just know that I was really concerned that 5 or 10 years, they're already asking people on the vineyard in Nantucket, do you want this? And it's getting written up in the New Yorker, and it's very sexy stuff to be working on, but nothing's happening. So I'm not really worrying about it right now, but it's hard to say what could happen in the future.
Fair
Yeah.
enough. Thank you.
Mm-hmm.
Just a comment, there's been tons of public engagement work in the Keys, especially around
Yeah.
G5 for Aedes control, and it's really fraught.
Mhm.
The public perception is really difficult to manage. So I think it's good to keep that in mind that if it was a pan pathogen gene drive, then I think it would be an easy sell. But if there's other Borrelia control tools, then it may be a
Yeah.
hard. I think it's going to take a lot of public engagement if they ever want to roll it out, but that's all doable.
Mhm. Yeah, good point. Tom, did you have, and you're muted right now. Yeah.
I just wanted to offer you're not seeing what the potential competition between Lone Stars and black-legged ticks would be if
Yeah.
even on a deer, but the concept that I think we're mostly thinking about is one of immune exclusion. So the deer bitten by Lone Star ticks after, for instance, when black-legged ticks would be there in the fall and the early spring, and then the Lone Star ticks would come out. And it could be that there's an immune exclusion phenomenon that's going. And there's been some little bit of work by Erica at Penn State that suggests that they definitely seem after Lone Star bites, the black-legged ticks don't do as well.
Really? So they don't do as
Mm.
well, the adults don't do as well on the deer after the deer have been bitten by them.
Sensitized by Lone Star ticks, yeah.
That would be fascinating. That would be a good thing for some diseases anyway.
Well, I
But
mean, on the
yeah,
other hand, it just means that the Lone Star ticks become predominant. So choose your poison, right?
I know. But so is that work published that you just talked about? Like I can't,
No, it's just she had one trial, and it was pretty interesting. The results were pretty compelling.
yeah.
But
Tom, do you know how long after the Lone Star infestation the scabs were put on? Was it
yes,
like a
so
reasonable?
it was a few weeks, I think, several
Gee,
weeks.
I guess it just raises the question if you'd see the same effect with the natural seasonal peaks of the Lone Stars
Yeah, I mean, I think
versus
actually
the
I may
scabs.
be misspeaking here. I just saw this not too long ago. We had a discussion between Erica and an immunologist and myself, and
Mhm.
so you'd think I could remember it better than I'm remembering now, but her point was to try to more or less mimic the the break in between the species. So the sensitization, I think, was at maybe 2 or 3 week intervals, and then the challenge was longer after that, I think.
Mhm.
Mhm.
That is fascinating. And you know, the densities of the Lone Stars on the deer can get to be so high they can actually kill a deer. And then in some places it's just really high density. So you could imagine there might be a response to the deer that can handle it. Mm, yeah. Any other comments? I'm glad we have Patrick Roden Reynolds is on this call, and also Leah Hamner, who's Leah. Welcome to you guys. I don't know if you want to say anything while we have time. We've got a little bit of time left, but good to have this group. And yeah, I'm so
Yeah,
that question. Oh, go ahead.
I was just saying hello. Working from my phone today. The Zoom kept crashing on my computer. I'm not sure what was going on. But
Oh,
yeah, looking forward to kind of looking more towards Lone Star and tick issues on the vineyard.
yeah, and this is Patrick who's in charge of those map data, which are I think really valuable. And Patrick, just if you haven't met Patrick, like I'm just going to say a few things. He has a lot of background in working on deer for his master's, deer and tick interactions for his master's in Maryland, and now being on the vineyard, knowing exactly which ticks are where and when. It's just Patrick's a great resource.
Yeah, it's interesting. You mentioned Erica from Penn State. I'm assuming Erica Mochtinger. She was one of my old bosses when I was in Maryland.
Yeah,
started on that. Yeah, USDA tick control project with deer, and it was an area-wide tick control project, and started as a field technician when I believe Erica was a postdoc then.
Wow, cool.
And I'll just hop on and say hello. Hi, everyone. My name's Leah Hamner. I'm a epidemiologist for Martha's Vineyard and also work with Barnstable County, Cape Cod. I'm really like an applied epi background, public health in action, kind of local public health background. So I don't have the luxury of specialization. I work on everything, but I do have a question for you, Allison. Thank you for a wonderful presentation. You've shared your results with the Chappie group, I believe, and I was just curious about what the reception was about. Did they find something to act upon, or did they find it just validating to their experience? Like, was there any sort of behavior change conversation?
Well, so in Chappaquiddick, they have an association of property owners, and they also have a committee of people working on the tick problem. And that committee is very excited that we're here, and they're hoping that they can kind of use our study as ammunition for anything going forward. I just basically validated everything they already know about their tick problems. I don't think there's too much new, but I can't speak for the residents of Chappie, but I think that our report, as it grows and as it gets published and as we work more with Patrick, will be helpful to them and to everyone on the vineyard as the baseline data mostly. Yeah.
Thank you.
Yeah, sure. And thank you for speaking up. And I hope I get to see you this summer too. Good.
Yeah, definitely planning on it.
Any other comments? One thing that I didn't show you that I'm trying to do, I'm really curious. So keeping me in mind if you ever hear of any interaction between lone stars and black-legged nymphs that would be relevant to what the questions we're looking at. I'm also curious, what's the minimum number of mammals that can support ticks, and what is the role of the cotton-tailed deer? I'm really curious to know more about that. And it turns out that there's an island off Martha's Vineyard called No Man's Land Island, where there are no mammals except muskrats. And the Fish and Wildlife Service has released New England cottontail rabbits there like 3 or 4 years ago, and they're reproducing very, very well. So I've been in touch with them, and I was going to go out there. I don't expect to find ticks, but I think it would be fascinating if there were lone star ticks reproducing on the rabbits. Maybe some of you have some ideas about rabbits, like can they groom most of the ticks off of themselves, or what is their role as a blood meal host for lone stars and for black-legged nymphs? I haven't found very many papers that really address that, even though they're common species. Yeah, Tom, and you're muted.
Yeah, so actually, I feel like Joe Peisman went out to no man's land years and years ago in the early days. I don't know if Steve remembers any of those stories or not, but in terms of rabbits, so they have their own tick, Ixodes dentatus, and
that might be there too.
They can have huge numbers of, I mean, their ears with dentatus look like those deer ears that you were talking about with lone stars. So I don't think they're particularly good groomers necessarily, because then they would be able to groom those ticks off.
Yeah.
But they're another example of developing an immunological resistance. So when we feed ticks on rabbits, black-legged ticks, they very quickly develop a resistance reaction to future feeding.
Really?
So yeah, they're not particularly good hosts
and
for
they won't do that
ticks.
with the dentatus, just the black-legged?
That's one of those probably species adaptations, right? So the dentatus have figured out how to not sensitize the rabbits. So it must be something they don't spit into them that black-legged ticks do spit into them.
So you wouldn't really expect the rabbits to have a big role in the black-legged tip life cycle.
No, but I would bet that with rabbits out there on no man's land, they probably would get dentatus.
Okay. Thanks.
Your question about, I don't know how you said it, the minimum animals or whatever. Mohican did just fine with white-tailed deer, Norway rats, and muskrats, and they had tons of Ixodes scapularis before they got rid of the white-tailed deer.
Yeah.
The rats and the absence of Paramiscus leucopus came into the settings and occupied the sites that would have been occupied otherwise by Paramiscus. I think the main reason you don't see Ixodes scapularis on rabbits is that the rabbits are mostly in the meadows and not so much in the forested areas. Whether that would change on a place like no man's land and the absence of other species, I don't know. But I would think that the driving thing is going to be that there's no deer there. That's what's going to keep
Right.
the scapularis
Yeah.
numbers.
Yeah, thank you. Yeah, and Mohican's like the perfect, so many things have been learned from those Mohican studies. I think that's really, I'm glad you brought that up. The rats taking the place of the mice. Okay.
Any other questions? Yeah, I'll just add that after Patrick and Leah introduced themselves and you alluded to the group, so there's a very interesting community of practice presence on the vineyard that makes it an exciting place to be doing this kind of stuff, because it's a very active place, as Tom knows, and Sam has been working there for a number of years, and so it's a good place for future studies. But thank you very much, Allison, and thanks everybody for coming, and we'll see you all again in 2 weeks.